Okay, here's the lecture for the scientific method, since it was not recorded properly. As you remember, at the beginning of class, I asked, I pointed at people about who were going to live and who was going to die. Most of you died, and I did that because I'm evil, I guess. It must be that. No, I wanted to point out to you that all of us are alive today primarily because of medical science, stretching back probably to, at least the 16th or 17th century. But things didn't really begin to change until the 19th century in terms of how long people lived, primarily because, an understanding of the germ theory was one thing. So, on we go. I gave some definitions of what biology is. Study of life is, of course, study of life, including life and living organisms. What is life, including their physical structure, chemical properties and processes, molecular interactions, physiological mechanisms, development, and evolution. So, what is biology? Well, we're going to define life at a point, but it certainly includes evolution, because it turns out that at least all life that we found on Earth, and it all appears to have been life only appears to have originated once, because all life organisms follow the same genetic code. And then phylogenetics, which is essentially just the evolutionary relationships between all living things, past and present. Ecology is interaction of organisms with their environment, including their abiotic environment, means just the physical things like temperature, if you're in the water, pH, salinity levels, percentage of oxygen, just the general physical environment. And then also organisms interact with the biotic environment, which is all the other organisms in their environment, including species that are the same as themselves, as well as other species. All life develops. We'll get to cell theory, genetics, homeostasis, all that kind of stuff. So, we defined in class, I asked, what is science? Study of all natural You're probably wondering what phenomena, all things in the natural world and the natural universe. Science is also a way to study the natural world. Of course, there's many ways to study the natural world. We have all these other disciplines like history and literature and religion and all these kinds of things. So what's this about science, and is it any better than any other way to find out about the natural world? And science, a specific process be systematic search Okay. Okay. Let's start this. So, imagine that you wake up and you don't know anything. You're lying down, you're staring up. Turns out you're lying next to a river. And it comes from a canyon to your left. The river comes all the way through the canyon, past you, into the straight plane going the other direction. And at the same time that you notice this, you notice there's this big, bright object above you. And it traverses the sky. Goes from the canyon, or the rim of the canyon where it first appears, all the way across the sky, following the river, and then setting exactly where the river fades off into the distance. And it disappears. And then it gets dark. You don't know what's really going on. But when it gets dark, you notice all these points of light above you. And you realize during the night that these points move all the way across the sky in the same direction as this bright orb did before. And there might even be a bigger white object, maybe only a quarter or a half of it, and it too follows the same path. And then finally, all of a sudden it starts to get light again, and that big orb rises up again from where the canyon is, 180 degrees from where it's set, and it too follows all the way across the sky. And again, it sets, the stars come out, and this goes on, and you wonder, where am I? So, what would you think if this is all you knew at this point about you were wondering where you were? Where do you think, how would you answer that question? Where am I? And I asked you that, and finally someone said, well, I think I was at the center. And so in the center of the universe, that everything seems to revolve around where you are. And that would be a completely intuitive sort of thing in terms of determining, oh, this is, I think, where I am. It's like an explanation. In fact, we would call it a hypothesis. That the hypothesis is that I find myself in the center where everything else is revolving around us. And that would seem completely intuitive. The question is that the true explanation of where we are? And of course, the answer, of course, is no. But at one point, everybody who might have thought about where they were thought, oh, we're at the center of whatever place we're in. We can call it the universe, the world, who knows, anything, but we're at the very center. Even though that means everybody thought that, but that doesn't make it correct or true. And it wasn't until many centuries later that someone, because of some anomalies, namely there are some points in the sky that instead of following this path, the same path all the time, they wander. And those, in Greek, that wandering, the name wandering or wanderer is planet. So that suggested, well, how do we then account for everything else is going around and around, but these planets appear to wander all over? And it wasn't until later, in the 15th century, that Copernicus came along and suggested that actually, instead of a geocentric, geo means Earth, centric universe where everything revolves around us, geo is Earth, it's actually a heliocentric solar system where the sun is the center of the solar system and all the planets revolve around the sun. And then, of course, beyond that, there's other stuff. But he came up with a model that didn't really work very well because he thought the planets all circled in a circular fashion when they actually follow an ellipse. And it wasn't until Galileo and Kepler and then Newton who finally showed and developed a model that predicted where the planets were going to be at all times, and also explained why all these other points of light are, and that white blob that comes out at night, well, it comes out during the day too, but not the sun, is actually the moon, and it circles the earth. Okay. In fact, it does actually circle us. So you can be in a situation where everybody thinks something is correct and they're all wrong. So science can actually get through that kind of a problem and actually figure out what is really going on as close as possible. So why do we do science? To figure out what not what might be going on, or you hope what going on, but what actually is going on. Someone said curiosity. And in fact, that's why we might want to figure out what's going on, because we're curious. We want to really figure out what's going on. And in fact, curiosity is what really drives science. There's another reason. It's a pragmatic reason. Science provides useful information, and some people think we do science for that reason, and it's true, but basic science, we're just trying to figure out what's going on, that's mostly driven by curiosity. And that's where all the great discoveries have come from. But science isn't just those great discoveries, it's the process of making those discoveries. And we do it in a specific fashion. But it's generally driven by curiosity and not for necessarily practical purposes. It can lead to practical purposes that make our lives better, but fundamentally, it's about figuring out what the world and the universe is like, because some people are curious. Okay. Now, we'll go through all this, but we'll just go to the bottom one. It says, second, how do we do science, and what is the scientific method? Okay, so this is the basis of science. This is Richard Feynman. He's a Nobel Prize winning physicist at Caltech. He's just giving a little talk to incoming freshmen at Cornell about what science actually is. Okay? And I'll discuss this in a second after we watch this.
Now I'm going to discuss how we would look for a new law. In general, we look for a new law by the following process. First, we guess it. Then we so don't laugh, that's really true. Then we compute the consequences of the guess to see if this is right, if this law that we guessed is right, we see what it would imply, and then we compare those computation results to nature, or we say compare to experiment or experience. Compare it directly with observation to see if it works. If it disagrees with experiment, it's wrong. And that simple statement is the key to science. It doesn't make a difference how beautiful your guess is. It doesn't make a difference how smart you are, who made the guess, or what his name is. If it disagrees with experiment, it's wrong. That's all there is to it.
Okay, so he's saying look for a new law. Well, we'll say for knowledge of some sort.
Now I'm going to...
Oh, and And he says something, let's say we see something, we want to know why it is. Like, let's say, why the sun comes up in the east, we call it is the east, and then it sets in the west, and then we come up with an explanation, and we make a guess. And I mentioned earlier in class about, I took my keys and I dropped my keys on the floor, and I said actually at one point in California, people believed there were other people with metal heads living in the center of the earth that accounted for gravity through electromagnetism. And if you remember or know, a magnet works only on metal, and they believe that everything sticks to the ground because of gravity is actually electromagnetism. Now, we could turn that into an experiment, or we can make a prediction. Well, if it's true, electromagnetism only works on metal, and so all you people in the room who are sitting down, you're not floating off from your seats. And so we can say that, obviously gravity is electromagnetism. Electromagnetism must be something different. Okay, so even something as simple as that, you can actually disprove or show that it's wrong. And this is the thing that Feynman is highlighting. Science, the great thing about science is you can be wrong, and you can learn about things by being wrong instead of trying to figure out what's right or proving what's right. We don't actually prove things in science. We mainly try to do something that's kind of counterintuitive, which is to disprove things. Now, if you have an explanation that's really good, and every time you try to disprove it you can't, then over time you get stronger and stronger evidence that perhaps it's actually approaching the truth or close to the truth. Okay, so being wrong allows science to learn new things. Because, by the way, every time you think you're right, usually when you're right and you found an answer, you basically stop looking. You think, that's the way it is, and that's it. And in science, there is no absolute truth. There's provisional. So there's provisional knowledge that is in fact basically fact, but if new information comes along that disproves, let's say, a prediction of that idea, then you can be wrong. It's not like it's dogma. Okay, so here's the scientific method. Really simple. We'll start with observation. We come up with a question. A hypothesis is nothing more than an explanation. A prediction. You test the prediction with an experiment or an observation. You come to a conclusion, either that your hypothesis is there's support for it, or that it's wrong, and then you have to go back to come up with a new explanation. So you go out one morning and you try to start your car, and your car won't start. And you think, okay, God, that's bad. I wonder what it is. Well, let's say it won't start. You ask, why won't it start? There's all kinds of explanations. The simplest one might be, the battery is dead. Perhaps you left the light on. Okay? So, what do? If the hypothesis is correct, you can predict that if you get a new battery and put it in there, or try to charge this battery, you should be able to start the car. Which you do. You replace the battery. If the car starts, then the dead battery hypothesis is supported, right? But if by replacing the battery the car still doesn't start, then you're back to square one. And so you have to come up, there's all kinds of different reasons why a car might not start. It might be the alternator. It might be the starter motor. it might be that rodents have gotten into your engine compartment and chewed up all the wiring. That actually happened to me once. So what is a hypothesis? I asked everybody. And some people said an explanation. For some observation. That's good enough. So where do hypotheses come from? They come from observing things initially. They also might come from thinking about things, theorizing. And we'll get into that a little bit later. So are these hypotheses? The universe revolves around the Earth. For, let's say, why we find ourselves where all the stuff, all the planets and everything revolve around us. We see that around us. It's because the universe revolves around the Earth. That's a hypothesis. It's not a true hypothesis. In other words, it's not true. We can disprove it, but it is a hypothesis. Is deciding what to wear based on weather a hypothesis? In other words, let's say you wake up in the morning and it's boiling outside. You know it's going to be very hot. How do you know this? You read the news, or you heard it on the news, or you poked your head out and it's early in the morning and it's already warm. So you think, okay, I better wear something cool. So you decide to wear shorts instead of a fur coat. How about you make a hypothesis, what's the best route to take to school? Let's say if you live in Burbank. You take the freeway at certain times, or do you take the surface streets, whatever. Those are all hypotheses. Actually, they're predictions about what might be the best route, and you come up with an explanation. Well, the freeway is probably faster because freeways generally are faster. You can go at a faster speed, but it might not necessarily be true at that time. The main thing is that you can always, any explanation you come up with, you can disprove. You have to be able to refute it or disprove it, or what we say often is falsify it. What do we mean by falsifiable? Well, it's simple. It means that we can disprove a hypothesis. If you can't disprove an explanation, then it's not a scientific hypothesis. Okay? Let's say you come up with an idea that spirits cause thunder and lightning, and they do it because they're angry. Well, we can't test two supernatural things, so it's not a scientific hypothesis. Okay? Now, we can certainly test and perhaps figure out where lightning and thunder and lightning actually comes from, and we have. And there's a perfectly natural explanation based on the laws of physics. Okay? Didn't involve supernatural things. So we can say, we can't discount the supernatural, that some mean demons out there are throwing lightning bolts around, but we can certainly say, for the most part, lightning and thunder are natural processes based on electromagnetic radiation and electric fields that are caused by things where I won't go into now. Okay? So, should we look for evidence to confirm a hypothesis? Or should we look for evidence to refute a hypothesis, an explanation? Or why would you want, in other words, to refute or disprove, let's say, your favorite hypothesis? You come up with a great idea. why do you want to go out there and make trouble and try to blow up your explanation? Because it's a really good explanation and it's really creative and all that. And what does it mean to be falsifiable? Well, first of all, most people go out and try to confirm their suspicions about how things are. That's generally what people do. Rarely, because they want to be right. People like to be right. Even if it turns out they're wrong, they still want to be right. Well, in science, if you're really trying to figure out what's going on and not what you hope is going on, or you dream of is going on or whatever, is you want to make sure you don't fall for things that confirm your pre-existing conditions. In other words, you're trying to avoid fooling yourself. So in science, we don't confirm things. We actually try to disprove things. So we take a hypothesis that we come up with. It's really creative, and we want to figure out the best way to show that it's wrong. And hypotheses generally make predictions that you can test. You say, Einstein's theory of gravity actually predicted, before black holes were ever found, he predicted that there would be black holes, but he didn't believe it himself. But that's what his theory of general relativity actually predicted. And in fact, it turned out to be true. But to do that, people figured out that's a prediction, and they went looking for black holes, and they're very hard to find, obviously, because no light comes from them. But you can detect them other ways, and it turns out we found actual black holes. In fact, it appears that most galaxies have a large black hole center of the galaxy. Okay, so people went looking for what the prediction says. If we had never found a black hole, it doesn't mean that they weren't there, but there's a good chance, perhaps, let's say we think we found a black hole. Oh, well, that's great. Is it really a black hole? And there's all these ways you can figure out the most likely explanation. With the highest probability is, in fact, there are black holes. So, these various predictions that, let's say, Einstein came up with, or Darwin in terms of evolution, or plate tectonics, or any hypothesis like your car starting, you can go and try to disprove those hypotheses. And if you keep coming up with information that supports your hypothesis, after really, really trying, you can say, well, provisionally, I believe this is the cause. So we try to disprove, and that's what it means to be falsifiable. A scientific hypothesis is not falsifiable unless it can be disproven. It's not a scientific hypothesis unless it can be disproven or is falsifiable. Okay. So scientific methodology, there's something called observation. Now, observation means seeing or measuring something outside you. Whether you can see it or not, you can hear it, there's some way that you can actually measure it. Empirical means, or empiricism means, so empirical means verified by observation or experience rather than theory or pure logic. In other words, we know that the interior angles of a triangle is 180 degrees. We don't have to go out. We can use that because of mathematics. We don't have to go out in nature and find it. Empirical evidence, on the other hand, you make predictions and then you test them in experiments like Feynman came up with, or observation if you can't actually go out and do an experiment. Like we'll get to those later. But that's what empirical or empiricism is. Testing observations. you can use advanced instruments or just your five senses. So usually we start with an observation, then we come up with a question for that. Why is that observation? And then we come up with an explanation or a model. A model is also sometimes used instead of hypothesis. And a model is comprised, or a hypothesis is comprised of a set of assumptions and should reveal predictions that follow logically from those assumptions. At some point in the process, the model can be put in the context of more general theory that connects many different models under a unified conceptual framework. So, we can have simple hypotheses, but they might be part of a larger group of phenomenon that, let's say, we want to explain, like the theory of gravity. And so, with theory, they're tested, these different hypotheses and explanations are tested over and over again, and eventually the models are refined, and they're not refuted, and after a certain point, you've got a theory that has a very strong evidentiary basis. And it explains some group of phenomenon as well as better than any other explanation out there. Doesn't mean it's completely right. It's provisional. In other words, it's the fact that in terms of understanding things at that point in time. Now, if a theory fails even one key prediction, it is falsified, and that means it's wrong. So, you can have a theory that might have been, or this idea of some knowledge that you thought was absolutely true, but some new information comes along and disproves it. That means that hypothesis, or if it's a larger theoretical process, it's out the door. You toss it. You have to start over. So no amount of experimentation can ever prove me right. A single experiment can prove me wrong. Is science, the process of science, robust to an incorrect hypothesis or theory? Yes, because you can prove something wrong. That means even if people believe something for 100 years or 200 years, it's not written in stone. Isaac Newton, was the first person to come up with an explanation for gravity. And his explanation worked very well. In fact, we still use his explanation and his mathematics to look for when comets are going to come by, or where we should shoot our spacecraft so that they intersect with the planet we're going to, things like that. But it turns out his explanation, his theory of gravity was incomplete or just plain wrong. And it wasn't until 1905, or actually 1915, that Einstein came out and came up with this general theory of relativity, which is a much, much better explanation for gravity and gravitational forces and how that affects the universe. And Newton's theory had stood for, well, since 1670. So 240 years, something like that now, 50 years. And, or until 1905, so about 200 years. No, 300 years, I guess. And it was discarded. Okay? Okay. So, here's a little video on the scientific method.
Impossible? We thought so. Unimaginable? We thought that too.
We count
You what?
like this. 1, 2, 3, 4, 5, 6.
Let's say this desert ant wants to go here for lunch.
Champity champ champ.
Because she's in the desert, there is no scent trail, so how does our little ant find her way home?
I know.
Well, some German and Swiss scientists think that ants have
pedometers in their brains.
meaning maybe there are brain cells inside a desert ant that record every step the ant takes as she walks out.
6, 7, 8, 9.
And then when it's time to go home, they just reverse and count backwards.
5, 4, 3.
It's a nice idea, but how do you prove it? Okay, scientists designed an ingenious experiment. Yes, which lets us literally see these pedometers at work. So we'll take an ant, we'll call it...
Ant number 1.
Okay, so ant number 1, we want to watch you take 10 steps to the food source.
1, 2, 3, 4, 5, 6, 7, 8, 9, 10.
And then 10 steps home.
10, 9, 8, 10, 11, 12.
Now, could you just go back to the apple?
Okay.
Thank you. Now, ant number 2. She also marched to the food source, but when she got there, they grabbed her and super glued little pig bristles. They're kind of like stilts. So now she's a much taller ant, and therefore every step she takes will be that much longer. Careful. Careful.
Is this absolutely necessary?
And finally, ant number 3. She also walks 10 steps to the food. Again, they grab her. This time they do something a little gruesome.
What do you mean gruesome?
We're cutting your legs off at the knees.
What?
Sometimes we've all got to make small sacrifices for science, so because your legs are now much smaller, from now on, every step you take will be much shorter.
I feel all stumpy.
Okay, now comes the fun part. When everybody was ready, the ants were released, off they go, and here's what happened. The stilty ant, she walked 10 steps home and stopped.
I'm out.
No, you're not.
What?
Because each of her steps is now so much longer, she'd gone right past the entrance.
What the?
The stumpy ant, she also walked 10 steps home and stopped.
I'm home.
But because each of her steps is now so much shorter, she only got part way back.
I want my legs back.
Sorry. And the last ant who was left alone, she gets home
I'm out.
with no problem, strongly suggesting that all these ants were somehow counting their steps. And just in case you're not convinced, that same day, scientists move all 3 groups of ants back into the nest for the night. And the next day, all the ants were marched off to get food using their new legs. The stumpies took lots of steps. The stilties took fewer steps. The regulars took regular steps. And when it was time to come home, all the ants, all 3 groups went straight home. Their pedometers apparently had adjusted to their new leg size. So ants, it really seems hard to avoid this conclusion,
I'm home.
do have pedometers in their heads, or another way to say it is...
Ant count. I want my legs back.
Okay. Now, things. We talked about sample size, and even though the video only showed one ant of each size, one regular ant, one stilty ant, one stubby ant, they talked about each group, and it meant that there were a whole bunch of stilty ants, a whole bunch of normal ants, and a whole bunch of stubby ants. like this. And that's because to do experiments, you want to have many independent points of data, and then you can make you can understand it in terms of a larger population, in this case a whole colony of ants or many colonies of ants. So what you have to do is you have to what's called set up an experiment with different groups, and it's not just one individual for each group but many, so you can get, let's say, a mean or an average number of steps for each group, because some might, obviously there might be some normals that take 9 steps or 11 steps or whatever like that, and stilties the same way, but they might converge on, let's say, 10 for the normals. And you want to make sure you don't just inadvertently pick one ant that can't count, right? So that's why you take what's called a representative sample. And the bigger the sample, the stronger your experiment. Furthermore, these were German-Swiss men scientists. Nary a woman to be found. Well, it turns out that there are many women scientists, and perhaps in the past they didn't get their due, but they do now. For example, there's this woman named Bunrell or Burnell, that's right, and she discovered about 40 years ago these pulsing stars. They're called pulsars, and they're actually neutron stars. She was a graduate student at the time. What she discovered, now she had an advisor and she showed him the data, and he said, oh, that's just an anomaly or something. But she was the one that actually figured it out, that it was a pulsing star from a spinning star called a neutron star. He got a Nobel Prize for it, she didn't. Okay? Even though it was really her who discovered it and should have. Fortunately, things have gotten better. There's this new gene editing technique that was first invented by a pair of women who are probably going to get the Nobel Prize this next year. So, the days of women being considered inferior are over. Thank God. There are great women scientists out there, just as there are great men scientists, no matter who you are or where you're from. Okay. This is another bit about, how we actually do things. Okay. What constitutes a scientific hypothesis? It's based on, well, we've already gone on this. But if your hypothesis is not falsifiable and testable, it is not a scientific hypothesis. And this is the thing: observation, hypothesis, the explanation makes a prediction, then you test it. What constitutes a good experiment? Okay. In general, when we think of an experiment, we think of an experiment where we're testing one thing with one variable. And we try to have an experiment that minimizes bias, either from the experimenter or from the subjects who are part of the experiment. And then we use a bunch of statistical analysis to determine if in fact there's a difference between, let's say, one group and another. So, let's say there's this idea that perhaps taking vitamin C protects you from getting cold. So, how do you test that? Well, ideally, you create two groups that are similar. In other words, if you look, ideally the two groups will be well matched. Average age of each group, they don't have to be exactly the same, but average. So you can create a dummy variable for age, let's say, or you can create, maybe there's differences in gender, so you create a dummy variable for different sexes, exercise habits, things like that. But the main variable, those are just the controls. The ones that you really want to look at is whether, let's say, vitamin C helps or hinders in terms of protecting you from getting a cold. So you take a bunch of people and you randomly pick, without foreknowledge, which ones are going to get a pill of vitamin C and which ones are going to get a pill that looks like vitamin C but isn't. It's placebo. And even though you have these other differences that might add kind of variation, you can control for them by just creating variables statistically that you don't have to know about. What's the name of the variable being manipulated? In the pen dent. Or treatment variable. Okay, so you want to find out about vitamin C. What you do is you have two groups. One of them gets the treatment variable, vitamin C. One of them gets, or treatment, they get the vitamin C. And the other one gets the placebo or control. Okay. Now, and then what's the name of the variable you actually measure that responds to the manipulation? Well, in this case, either they get a cold or they don't. So that's called dependent or response. Okay, so I asked you this question before. Is it better, here's the hypothesis. Vitamin C causes increased resistance to catching colds. In other words, it protects you from colds. Is it better, what's the best process or setup? Do you take a survey and ask who takes vitamin C and how often they catch colds and see if there's a relationship between vitamin C and cold resistance? And that's one way. Or do you do a survey to ask who takes vitamin C, then put the vitamin takers into one group and the rest who don't take vitamin C into another group. Then you exposed each group to a mild virus, a rhinovirus, and see who catches the cold, noting the difference between the two groups. Or finally, do you take a large group of subjects and randomly divide them into two groups? Then have one group take vitamin C supplements for 2 weeks without knowing they're actually getting vitamin C. They get a tablet that they assume is vitamin C. And the second group takes identical tablets with no vitamin C. It's a placebo for 2 weeks. And then you expose both groups to a mild virus and see who catches the cold and note the difference. So you're supposed to say C. And the important terms here are random. You want one overall population, and you want to sample from that population, and you want to randomly pick each individuals for either getting vitamin C or not. Okay? You don't want to say, oh, this one goes in vitamin C, this one goes in vitamin C, this one goes in vitamin C because he's handsome, whatever. No. They have to be randomly picked. Okay. Now you also, of course, need a good control group, meaning a control group where the subjects think they probably get the treatment, but they don't know. In fact, no one knows. You can tell the people up front. You can't tell them what group they're in, but you can say either you're going to get this drug, in this case vitamin C, or you're not. But you won't know the difference. Because there's something called the placebo effect. You don't want to measure the placebo effect. You want to measure whether, let's say, a drug works or not. And if it's a placebo effect, it's like a hidden variable. And placebo effects are real. They have real consequences. So there's subject or volunteer bias. They have to not know which group they're in, but that's not always possible. Example: you want to find out how much a person's reflexes are affected by alcohol. So, one group you give alcohol, let's say bourbon, 2 ounces of bourbon. And then the other group, you give a colored liquid, but it's not bourbon, and it actually doesn't have any alcohol in it. Well, the group that is actually taking the bourbon will know it, and the group that isn't will know it too. Okay? So you're not really testing reflexes because of the placebo effect. The people who aren't drunk already know they're not drunk. Okay. And then there's experimenter bias. Not obviously, but experimenters might want their drug to work. And so they might look to confirm that their drug works. So they cannot know what individual is in what group. There will be a list that they can look at after the experiment, but they want to know for sure. Okay. Okay. Do you tell subjects which group they are in, whether, let's say, they get a drug or don't get a drug? No. What is it called when they don't know? A blind experiment. How do you treat the control group? The same way you treat the treatment group. If one gets a drug in pill form, the other gets an identical pill without the okay, this is filled in. Okay. And experimenter or technician bias. Why should the experimenter not know which subjects are assigned to which group and which group gets the treatment or placebo? It's because of confirmation bias. In other words, if you come up with a new drug, you want it to work, but you want to make sure you're not fooling yourself. And you want to find out whether it really works or not. And so, when you assign the subjects, the researchers cannot know which subject is actually getting the drug and which one isn't. They will have a file that will match, so Mr. Smith is in 1A. He's getting the drug, but they can't see it. Mr. Smith could have been in the drug group or the other group, one or the other. It's randomly assigned. What do you call an experiment in which neither the subjects nor the experimenter know? It's called a double-blind experiment. That's the gold standard in science. Well, instead of measuring the whole population, you want to use a smaller sample to make it manageable, but still know or understand enough with the results that you can then apply it to the whole population. That is what statistical analysis is. We often get certain parameters from that, like the average. So, let's say with taking alcohol, if you drink 2 ounces of bourbon versus 2 ounces of water, it might take you half a second to react, to hit the brake, versus only 1/10 of a second. Okay? So you get a bunch of individuals. Of course, in this case, we've got a subject bias problem, but we can still handle that. So we'll get an average time. The average time for 100 people who are drunk may be half a second. Some may be faster, some may be slower, but they average half a second. And then the same thing for the undrunk group. And then you also want a measure of the variation around the average. So you want to find out how big of a spread there is, and there's a thing called the standard deviation. Okay, so sample size matters. Sample size, the number of subjects tested in each population. Usually you want a larger sample if possible. So let's say you'll never, obviously you don't never want one individual for each sample or for each group. So let's say you want 10 individuals who are getting some drug and 10 who are not, or 10 who you're testing 10 to see what the reaction time is on drinking like 5 shots of bourbon compared to not drinking any. Okay, reaction time in terms of how fast you step on the brake. Okay. Or do you want 100 individuals for each group? Well, there's another thing called the effect size, and that's the magnitude or the size of the actual effect. So let's say we have an individual, or we have 2 groups, and they're going to get, one's going to get bourbon, and one's not going to get bourbon. Now, if the bourbon group only gets 1 shot of bourbon, they're not going to be very drunk. And the reaction time, let's say the normal reaction time for someone who's not drunk is 0.1 seconds from the time they see something until they step on the brake. Okay? And we're testing this in an experimental setting in the lab. And now, if you give someone 5 drinks, 5 shots of bourbon, they're going to be really drunk. And we might say that if we tested a whole bunch of them, the average time it takes to react might be 0.7 tenths of a second instead of 1/10 of a second. Okay? That's a large effect size. If we only had given the alcohol group 1 shot of bourbon, the effect size for them might have been 0.2 of a second versus 1/10 for the group that didn't have the alcohol. So in that case, the effect sizes would be very small. There might have been a real effect, but you need more sample or a bigger population to actually detect it. Whereas with a big effect size, let's say 5 drinks versus no drinks, 0.1 of a second versus 7/10 of a second, that's in a big effect size. You only need, let's say, 10 individuals for each group. Instead of the other one, where each person who gets alcohol gets 1 shot, 1 ounce of bourbon, then you might want 50 in each group. Okay. And then you want to come up with some statistical measurements. The most common ones are the mean or the average of the sample. So for example, like I wrote like here, 1, 2, 1, 1, 2. If you add all those up, you end up with 7. And then if you want to get the average, you have to divide by the number of individuals, and there's 1, 2, 3, 4, 5 individuals. So the mean in this case is 1 point something, 7 divided by 5. 1.3 or something like that. Okay? So that's the mean. Now, the standard deviation is actually the spread of from each individual. They're not all going to be at, let's say, the bourbon group will not all be exactly at 7/10 of a second to react after drinking 5 shots of bourbon. Okay, some are going to be at 6/10 of a second or 5/10 of a second. Some at 1.2 seconds, right? And so the standard deviation will capture that variation around the average. And then what we look for in terms of statistics is the probability that 2 samples are from different populations. Namely, let's say we're looking at the effect of alcohol. We're actually now we're picking individuals randomly from an overall population, but then we're treating them differently in subpopulations. So we have the bourbon population and we have the placebo population. And that way we can find out, is there a real difference? And you can mathematically figure this out depending on the sample size and the effect size and the standard deviation. And the smaller the effect size, the bigger the sample you need to detect the difference. And of course, obviously, a large sample size and a large effect size increases the probability of detecting a difference between the population samples. But, like I said, if you have a really large effect size, you don't need such a large sample size. If you have a really small effect size, let's say between 1/10 of a second and 2/10 of a second, then you need a much larger sample size to detect a difference. So why is sample size so important? As I mentioned, with a large effect size, you can get away with a small sample size. But with a small effect size, let's say if we're talking about alcohol consumption, so let's say if you've only had one drink of bourbon instead of, let's say, 5, you're going to be much less drunk compared to someone who hasn't had any drinks. And so the effect size is going to be much smaller. So if you have a small effect size, you actually, if you're going to detect any difference in reaction time between effect size, between drunk, a little bit compared to a lot, you're going to need a much larger sample size. So here we have this the statistics, and in the first 2 columns, right here, we're flipping, let's say, a coin, and obviously, if a coin is, it's 50% heads, 50% tails. So if you flip a coin 10 times, you should be close to 50%. But obviously, if the first time you do it for one group, you might get 6 heads instead of 5. And then the next time you might get 5 heads, and the next time you might get 5 heads, and the next time you might get 5 heads. Right? So each change from one digit to the next is 10%. Now, you could also end up, let's say the second group is flipping, same thing, and the first person who flips only gets 2 heads. Now, the probability of that happening is much smaller, but it can happen. And then he flips it again, or the second person flips it, they get 7 heads. And then finally, there's 5 and 5, as would be expected. But each single flip is worth 10% difference. Now, as we saw here. But if we flipped it, let's say, so there were 10 times, here's 100 times, okay? And it's being much more precise. So a person in group 1 flips it 100 times and they get 61 heads. The second person gets 53 heads, then 55, and then 51. We're now talking about, instead of 10%, only 1% change. In other words, it's much more precise if you do it 100 times. Okay, so 61, 53, 55, 51. And group 2, 44, 65, 52, 57. Right? Same thing. Now, if you do it 1,000 times, you'll get even more precision to detect actually a difference. And it'll be, let's see, 1%. 1/10 of 1% a person flipping it 1,000 times. So the first person might flip it 1,000 times and get 612 heads. Another might get 449 heads, 535, 651. Okay, and then we can actually see some differences, how much more precise things are in terms of right here. Actually, I'll look at this one. So, hold on a second. Okay. So, this sign right here is the sum sign. So if you add up all these numbers, 6, 5, 5, 5, you get 21. And then group 2, 2, 7, 5, 5, you get 19. The mean number of heads for 4 individuals flipping it 10 times in each group is 5.25 compared to 4.75, but there's much greater variation. So right here is the mean, the average, and right here is the standard deviation. In other words, the variation around the mean. And you can see with the first group, it's almost all 5s, except for 1. It's 6. But in this one, there's a lot more variability, so the standard deviation is much bigger. But we get much better estimates the more times we flip. And in terms of precision, so 55.5 is the mean out of 100 instead of 5.25, which if we did it 100 times, that would be 52.5 versus 55.5 or 54.5. So much more close in the group of 100. And then there's even more precision in the 1,000 times. And so the average is 554.25 versus 550.25. Okay? A difference of 0.4%. So when you read something, let's say they give you numbers, and it's only a very small sample size, let's say a sample size of 10 for each group. If there isn't much of a difference, you have a very small chance of actually detecting it, and you also have a larger chance of getting some subject is a little strange, and that will have a big effect on the overall numbers. But if you have a very large sample size, it will all even out, and you'll have a much more precise average time. Let's say in this case, average times you flip a coin 1,000 times, you're going to be much more closer to 500 than some individual who only does it 10 times. Okay? So, and that way you can actually detect a more precise difference. Let's say the effect size is only, if you've got 1,000 people doing the drunk test, and that one group gets only 1 drink and the other group gets no drinks, you will probably be able to actually detect the difference if you have 1,000 subjects in each group compared to only 10. Okay. Now, so we want to know, is the difference statistically significant? And so here we have vitamin C consumption and number of subjects with colds, right? So it looks like those who got no vitamin C, 90% of them got colds, right up here. Okay? And those that got 1,000 milligrams one of them got colds, 10%. Okay, so you want to know, is this different statistically? But the thing is, if you only have, let's say, 4 individuals in each group, the actual error bars, and this is what you want to see in terms of standard deviation, are going to be very wide. Okay? And so, in fact, very wide. Let's see, give me this. Okay. And here, you might not be able to actually detect a difference because there's an overlap here, right around here, right? Now here, it looks like 60% versus 40% or 50%. Is that statistically different? Well, if let's say you actually had 1,000 participants, this might actually be a statistically significant difference. So every time you let's say see something in the paper about 60% of the respondents said this or whatever, it depends. The bigger the number of respondents, let's say, the more precise your estimate of what the average response is, okay? And the more likelihood you can actually see that there's actually a difference. Okay. So, let's go over this again, just quickly. A well-controlled experiment. You only want to test one variable. You want to minimize bias by either the subject bias, like do the subjects actually know they're getting a drug, or is it hidden from them? And then the sample size and statistical analysis to see if there's really a difference. Okay? Now, the trouble is that you can't always do a well-controlled actual experiment. For one thing, not in a controlled environment, let's say where you're doing an experiment in an uncontrolled environment where everything is identical except for the variable being tested, unlike a lab. So I wanted to determine how well animals cached seeds. Okay? Now, the thing is, I could have done it in the lab with kind of an 8-foot long contraption where I could put rodents in and seeds, and they could bury the seeds, and everything would be very controlled. But the thing is, they don't necessarily, let's say, rodents often bury seeds to hide them from competitors. But they don't often act normally in, let's say, an environment in the lab. So I want them to have a more natural environment where they're more likely to behave more naturally. And so instead of actually, let's say, doing a trial with these rodents that I was working with for my dissertation, we built these big enclosures, 15 meters by 15 meters on a side, and they were sunk down so if you put rodents in, they couldn't dig themselves out or anything like that. And then we actually put the rodents in this much bigger space where they will act much more like they might act in the wild. And in fact, this is a location where I actually I trapped the rodents there, so they're already familiar with the area, and I could actually get much more, let's say, natural behavior. The thing is, of course, when I ran the trials, there were differences. There are different temperature differences, because I couldn't run all the trials like I could in the lab in controlled conditions. I had extraneous variation, but on the other hand, the rodents were actually acting more normally in terms of behavior. And so, in some sense, I probably got better data, even though there might have been differences of where they were acting because of, let's say, temperature or the time of day or things like that. Now, I try to control those kinds of things, but, we're in a lab in one case where the temperature is perfectly controlled, and in the other case, we're outdoors, okay? And then, there's a situation where there's uncontrolled variables because of bias. So like I was telling you about looking at the effect of alcohol consumption on driving and reaction time. In an experiment testing the effect of alcohol on that, those in the treatment group drink 2 ounces of 80 proof bourbon. Those in the control group drink 2 ounces of watercolor to look like bourbon. The trouble is, you can't control the placebo effect, because the subjects are definitely going to know which group they're in. The bourbon group can taste the alcohol. Even if you try to disguise it, they're still probably going to be able to determine that they're actually getting alcohol versus the non-group. Okay? So, you can't always do an actual well-controlled experiment. And that's unfortunate, but that's the way it is. Okay? So, the thing is, you can still run the experiment, and if you get a large enough sample size and randomize, also, you might, let's say, run each individual subject 2 times. Once where they're getting alcohol and once where they're not. Okay? And you vary it randomly in terms of subject. Subject 1 might get alcohol the first time, no alcohol the second time. Subject 2 might get no alcohol the first time, alcohol the second time, and so on, right? And so then you've got 2 measurements, and you can actually control a little bit for the fact that individuals, they know what they're getting, but we can individually see the differences in how the reaction times are. So there's certain things you can do to counteract those effects of the fact that you don't actually have a real placebo. And then, finally, there's a lot of problems, let's say, as I mentioned, that an asteroid hit the Earth and supposedly wiped out all the dinosaurs. And this was proposed from observation about iridium, which is an element that's found much more likely to be found on asteroids than on Earth. But there was this iridium layer that has been found in the same layer of rock from 66 million years ago at various places around the Earth. And a father and son team discovered this, and they suggested, and 66 million years ago is when the dinosaurs disappeared from the fossil record. So older rock, deeper rock, there were dinosaur fossils, and younger rock, younger than 66 million years, there were no more dinosaurs. Okay? So the thing is, of course, you could make predictions if it was an asteroid. And one of them that is that you should be able to find the remnants of an old impact crater, a very large one. And then a number of other things. And they did that. But the best thing to do in the best of all possible worlds, is that we would replicate the Earth, let's say 100 times, and 50 Earths would get impacted by an asteroid, and we'd see how many times, let's say, the dinosaurs got wiped out, and then you'd have another 50 Earths where either nothing happened, or let's say there's a dispute, well, was it actually an impact crater, or was perhaps volcanism, large volcanic eruptions releasing lots of toxic gas in the air? Maybe that was actually the real cause. Okay? So it would be nice if you could actually do that kind of an experiment, but you can't in historical studies. Okay? So in those, all you can do is go out and measure things in nature. You can still make predictions, like in the scientific method. You have a hypothesis that makes certain predictions, but you can't actually run experiments. But that doesn't mean that you can't do science. Because you can still make predictions. Maybe the evidence isn't quite as strong as if you could actually do scientific experiments, but still the evidence might be strong enough to give you an idea of what might have actually happened. So, what's a theory? In class, I asked people what a theory is, some explanation for some phenomenon like a hypothesis. Someone wrote an unproven explanation more like an opinion or an explanation that has been tested many times and has never been disproven. Okay. So, first of all, a theory is like a hypothesis, except it's usually some explanation, a larger explanation that ties in a whole bunch of different things concerning some phenomenon, instead of individual hypotheses about how certain things work. Now, of course, a lot of people say, let's say about the theory of evolution. Some people apparently don't like the idea that we shared our most recent common ancestor with chimpanzees, perhaps 10 million years ago. In other words, we're descended from a common ancestor 10 million years ago with chimpanzees, and they want to think that they're special as humans compared to chimpanzees. Okay. And so, in terms of evolution, they say, well, it's only a theory. In other words, hold on. Oh, come on, forget that. Okay, an opinion. So, that's basically an opinion, or an unproven explanation. It's only a theory is basically the opposite of a scientific theory. A scientific theory is a whole different animal in terms of what we're actually looking at in terms of evidence for something, some phenomenon. Okay? So a theory is a comprehensive explanation for a broad set of observations that is supported by a vast body of evidence that has been extensively tested and has not yet been proven wrong. It doesn't mean that the theory is correct, but it's been tested so much that as far as current evidence goes, that's our best idea of the truth of how something works. Like, for example, instead of the humoral theory of disease that the Greeks had, black bile, yellow bile, blood, and what else? And water. There's actually microorganisms that actually cause disease, and not a balance about these humors. So this germ theory of disease, which was formalized in the 19th century by a German and a French guy, Edward Koch and Louis Pasteur, and some other people, said, in fact, that microorganisms are responsible for diseases, and then they set out to show that that was actually the case. And so in the germ theory, to actually make the case that a specific type of organism is responsible for the disease, the organism has to be isolated from a patient. It has to be grown in a lab. That organism then has to be taken and used to infect another individual. Now, obviously you can't do that with humans, but you can do it with animals. And some animals, in fact, get the same sorts of diseases as we do. And so that's what they did. So there's a protocol that shows that if a bacteria or virus or something is responsible, this is how you actually prove it. Einstein's theory of general relativity is another theory. It's a theory of gravity. It's in fact, it's a much better theory than Newton's theory, which was still very good. But it's completely different and much more representative of what appears to actually be going on. The theory of evolution, or the theory of plate tectonics, where the continents move around on these big plates, and even though it seems like the continents would never have moved, but they actually have. So these are real scientific theories that have been tested over and over and over again, and they have not been refuted. So, hypothesis is a possible scientific explanation of an observation or set of observations that is falsifiable and testable. A theory is a comprehensive explanation for a broad set of observations that is supported by a vast body of evidence has been tested many times and in many different ways in terms of testing the predictions. That has never been disproven. Theories are often composed of many different hypotheses that explain related phenomena or a close related class of phenomena. Just a theory is a layman's explanation of something. And it's kind of the opposite of a scientific theory. It may or may not represent some true phenomenon, but it's unsubstantiated and more of an opinion than, and it's not rigorously tested. Okay. We talked about this in class. I made some jokes. Now, Dr. Feynman, I showed the video earlier, and he talked about laws, and sometimes laws and theories are used interchangeably. But in general, a scientific law is a verified description of an observed phenomenon, but it doesn't tell us why it happens. It just says this is what we generally see over and over and over again. A theory explains why something happens. It actually gives an explanation for the mechanism that causes that thing to happen. Okay. Evaluating scientific information. Obviously, scientists have to get their ideas out there. And so usually they publish in scientific journals where their research is written up based on theory and then experiments to test the explanations. And it's usually refereed by other experts in the field to see that they actually did what they said they did. And that's actually called primary literature. That's not the kind of stuff that you normally see in a newspaper. It's never the kind of stuff that you see in a newspaper. So primary sources involve original data written by the researchers who did the work. They're peer-reviewed. In other words, they're reviewed by other experts in the field, and then they're published. And this is how scientific discoveries are actually presented. And most people who read them are other scientists in that field. Now, most of you will be reading or hearing about secondary sources, either in magazines, newspapers, on the radio, on TV, and the internet, and they're based on primary source literature, but they're usually written or presented by a non-expert and summarized so that the general public can understand it. Now, there's been a philosophical debate about how science progresses. And we know a lot more than, let's say, the Greeks did. Or we know a lot more than, let's say, 17th century Europeans knew about, let's say, the natural world that we all live in. Okay. And there's been progress. In other words, our knowledge has increased over time. And the thing is, how has knowledge increased? And one hypothesis is a continuous model. In other words, you learn a little bit over time, a little bit more, a little bit more, a little bit more, and over a long period of time, you finally gain a much better understanding of the world and certain phenomena that are in it. And it's kind of like those who are currently doing science are basically standing on the shoulders of giants who came before them, who each did a little bit and increased our knowledge. Now, the thing is, though, there's also evidence of what's called a paradigm shift. And that paradigm shift, oftentimes someone comes along who has an extraordinary effect on our understanding of some phenomenon. Not just a little bit, but completely overturns the field. In other words, it creates like a scientific revolution. The germ theory of disease was one such event, but at the same time, there was a lot of incremental understanding of it, but eventually it overturned the humoral thing completely and had a profound effect on human health. A better example was that Einstein came along and reformulated our understanding of, let's say, gravity. Newton had done a good job, but Einstein showed that gravity from a completely different perspective, and his explanation and the predictions that it made was much, 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 much closer to reality, and actually very, very different from Newton's. Most general people could understand a Newtonian idea of gravity. Einsteinian general relativity is a whole different thing that makes some very strange predictions that we're not going to go into. So occasionally, someone or some group might come along and really change things with a profound discovery. And then finally, I talk about empiricism. That means referring to the world or experiment or experience when testing predictions. The thing is, we're limited by our senses into what we can see and hear and smell and feel. And so our normal senses are limited into what we can actually discover. So we look up the night sky. We can see little points of light with different brightness, different colors, but we really can't see specific features. Well, when the first telescopes were made, suddenly you could actually see individual planets, and you could also see little moons circling those planets, like Galileo did in the 16th century. And being able to actually see that helped change the conception of the solar system. This was around the time of the revolution of Copernicus, who suggested that the sun was the center of the solar system and not the earth was the center of the universe. And he also, Galileo also showed the rings of Saturn, and the same things happened in terms of very small things. So this was a larger technological advance in terms of optics. In fact, these were the first modern optics ever made in terms of microscopes, telescopes, and also glasses for helping people see. And because we could, let's say, then see things that were very small, we could finally realize that there were actually little single-celled organisms all over the place, and that our our own body was made up of cells, and that was a huge change. So sometimes technology provides us with new information that we couldn't have detected otherwise. And nowadays, of course, we have all these new technological advances for measuring things and detecting things that we didn't have before. And so I asked, can technology change the questions we ask? Well, certainly. If you can suddenly see things that you couldn't see before, you can either ask specifically, why are those things there, versus you didn't even know they existed. Okay. Okay, so I want to talk about correlation. Correlation is an observed relationship between two variables, two groups, events, conditions. A really simple one is the association between weight and height in animals, or let's say in humans in particular. Obviously, there's a positive relationship. If we were to draw it, and we have height on the bottom and weight, so the x-axis, this is the x-axis, and this is the y-axis. We would see something like this. Let's see, we'll say from 20 pounds, 40 pounds, 60 pounds, 80 pounds, 100, 120, etc. Right? And as you go up in height, there's usually a gain in weight. Does height cause weight to go up? Or does weight cause height to go up? No. They're both measures of size, just different measures, and so they're correlated, but one does not cause the other. They're just different measures of the same thing. Okay. So, in this case, we'll say condition A often coincides with an increase or decrease in condition B. So you can also have an inverse relationship. Like this, where increasing, 10, 20, 30 goes down. Obviously not weight, but you can have things like that, negative relationships. Okay. So, in this case, condition A often coincides with an increase or decrease in condition B, or people who A are more or less likely to B. Okay, so this is the X. Oops, let's take this. Correlation and causation are easily confused. As I mentioned, let's say in weight and height, one does not cause the other. They are correlated and associated positively because they're both measures of size. So, here's one. People have been saying for a long time, let's say, that violent movies cause people to be violent. Or something like the popularization of rap music in the late 1980s coincided with a sharp rise in narcotics use and violent crime. A. Songs about drugs and violence point to B. Increase in drug use and violent crime. Now, there may be an association between the two, but the songs and drugs about violence actually increase drug use and violent crime. Probably not, actually. In other words, music about crime can cause criminal behavior. But what about the opposite? Increase in drug use and violent crime might increase songs about drugs and violence. It could go that way, or it could go both ways, for that matter. They both feed off each other because they're kind of, in a sense, both part of the same cultural background. But they don't cause one another, as we would say, cause and effect. Now, and then you can also have a situation where there's a seeming association, but it's actually caused by a hidden variable. So, people who are regularly wear seatbelts are more likely to be diagnosed with cancer than those who do not wear seatbelts. In other words, if someone came along, might say, "Oh, seatbelts cause cancer." Well, I mean, that's craziness. And in class, I asked this, and some people said, "Well, perhaps stuff in seatbelts are carcinogens or things like that." Well, no. There is in fact an association between seatbelt use and incidence of cancer. Okay, but let's think about it. What are the seatbelts for? They're to protect people from getting hurt or dying in car accidents. As far as their purpose, how does that affect lifespan in people? Well, what does it do? It actually increases lifespan. So you wear a seatbelt, you are less likely to die in a car accident. Now, a longer lifespan, when do people get cancer? Well, they can get cancer anytime in their life. Now, the longer the lifespan, the more opportunity people actually have for getting cancer. The seatbelts aren't causing cancer, they're just extending life. Furthermore, the older you get, the more likely you are to actually get cancer, because older people are more susceptible to cancer because their immune system isn't working as well. So, seatbelts, here it looks like, you wear seatbelts, you live longer. Living longer makes it more likely that at some point during your lifetime you will get cancer. Do seatbelts cause cancer? Well, of course not. No, in fact, there's no correlation. If you control for age, in other words, if you put a variable in age in there, the correlation goes away. Age is responsible for a greater number of cancers. It doesn't cause cancer. It just means that you have more time to actually develop cancer during your lifetime. A correlates with B, right? There are many possible explanations. Let's go back. So, if you actually want to show cause and effect, let's say you find a correlation between, let's say, smoking, cigarettes, and lung cancer. There's an association. Okay. Someone might ask, does smoking cause cancer? Well, there's a correlation, so you could actually do an experiment, though. You might first expose, let's say, rats to tobacco tar or tobacco smoke, and seeing if a group of rats exposed to, let's say, carcinogens in tobacco smoke are more likely to develop cancer than rats that are not exposed. That means you're actually controlling two variables, and you can actually show cause and effect. Furthermore, you can they've actually demonstrated the actual mechanism that certain alkaloids in tobacco tar that actually cause changes in cells and the genetic code, and then that leads to mutations and cancer. Okay. Oh. Now, what about anecdotal evidence? What is anecdotal evidence? Well, it's kind of like hearsay. Oh, like my grandma believes that drinking a glass of wine in the evening has prolonged her life. Okay, she's dead now, but let's say she believed that. Is this true? Well, no, but it might be. There might be a suggestion from an old folk tale that moderate drinking actually is healthy, or something like that. So it's just kind of like a wayward data point. It might relate to something that's true, but it's never been tested, and there's no real evidence one way or the other, let's say. So you could set up a situation where you could actually test that. It's the starting point for a hypothesis, right? Well, it says that echinacea doesn't protect against colds, but my Aunt Mabel takes echinacea every day and has never had a cold in her life. That's an anecdote. Okay? The hypothesis is that echinacea may protect against colds. My Aunt Mabel takes it and says she's never had a cold, but that's not a data point. We'd actually have to test it scientifically. And that's that.